Hola. Thank you for reminding me, Akshay. So, any questions before we proceed? All right. So maybe 2 minutes description about yourself. Your degree, maybe a skill or something that you want people to know about, or you play music or whatever, or you're an artist or you're good athlete, whatever. Just tell us a little bit about yourself so we can understand better who we are. And Himanshu, we like to start first.
Hi guys, I'm Himanshu, yeah, and I'm doing my master's in electrical engineering with specialization in robotics. So this is my last semester, and what I do a lot. I do watch a lot of movies, so I like movies. So that's, I think, if that counts.
Any hobbies? Any other?
Yeah, that's what I watch a lot of movies. I like movies.
Okay.
Yeah.
Yeah. Well, maybe you want to record your brain activity when you're watching a movie.
Yeah, I will try that.
Versus something else or different type of movies.
Yeah, I was,
You know?
while you were telling that, I was thinking of that. Okay, I can record that while watching different movies. So yeah.
Right. Yeah, I'm actually watching with my kids Lost in Space. I don't know if you have seen that series. Well, they
Yeah,
had the
I've heard
very
of that,
old.
but I did not see it.
It's in Netflix. It's the very old one, almost a comic, right? And then the high tech version today. It's very interesting to compare this thing, right? Luciano.
Yeah, hi. So I'm Luciano. I'm also doing master's in electrical engineering, and I'm usually very interested in neuroengineering research. That's what got me interested in this course. I'm doing research with Dr. Rose Rahid, and there are some projects related to using this Muse headband. So I got interested in learning how to use it here. Um, I I wouldn't say I'm a very creative person, but I have a guitar and I tried learning it a little bit. So I might try learning it while I had the headband to see what's gonna happen.
Great. Thank you. Pohita? Is that the right way to pronounce your name?
Yeah, it's Pujita.
Pujita. Okay.
Yeah. So I'm Pujita and I've joined a master's in computer science and this is my first semester.
Ah,
And
welcome to
yeah,
UH.
yeah, thank you. And
Great.
my hobbies are I also watch movies and I like to play sports. Specifically, I like playing table tennis.
Oh, yeah, that might be interesting also to try with the headset. Yeah.
Yeah.
Thank you, Sankal.
Hello all. So I'm Sankalp Parekh and I'm a master's in electrical engineering student and I've been involved in research under Professor Rose. And I also worked on seizure detection with one of your student, Alexander Steele, under the guidance of Alex that's present here. And I've also worked on creating control algorithms for containing stress detectors using skin conductance.
Great.
And I like playing chess.
Oh, good. Good. One of my kids, he's really good. He plays a lot of chess, going to tournaments. Well, you could record that. That's an ideal task to record brain activity because you don't move a lot, and you're really focused. Really very interesting to see how your alpha waves
Yes.
grow. Yeah, good. Well, thank you for joining us for this workshop, Christian.
Hi, good morning. My name is Chris, and I'm a master's in biology here at the University of Houston. I've been a student here for a while in my undergraduate. I was actually a biomedical engineering student, and I was also a member of Pepe's lab. So now I'm back here. I actually have a Muse headset with me that was loaned out a while ago. I've been using it time to time. They have a school meditation thing, another sleeping option. And this project was a great way to test it out in like a live action setting. So glad to be here.
Thank you, Chris. So Alex?
I'm a Ph.D. student in Pepe's lab. I work on, well, two things mainly. I work on a stroke rehabilitation system by predicting movement intent, and then I've applied a lot of deep learning to different EEG tasks. I also play a lot of chess, so it's good to hear because I'm going to be.
No, we can organize a tournament here within the group, and have two. It's interesting because when you have two brains interacting, in this case through the game of chess, that you can start looking at brain-to-brain communication and synchrony, and phasing. It will be very interesting. If you get together and play. You could play against each other. You can play against a machine and then compare.
Yep.
This. Maybe we should do. We should do a workshop just on chess and I play chess, but I just play with my kids. But now they win all the time.
Yeah. Young kids who go to tournaments really kind of scare me a lot. So yeah, I'd be interested.
Yeah. Oh, Anna.
Bidding your true value in a second price auction.
Chase, I think your audio is unmuted. Yeah.
Oh, okay. I thought it was his turn. Okay, Anna. Okay, hi everybody. My name is Anna. I think I'm the only one who is not pursuing a master's degree here.
That's okay.
And also the only one who has zero experience in neuro, But my interest for this came with, I don't know if you guys have watched this new movie, Upload. Upload is a new movie in Amazon Prime. It explains how the brain works, and it's just amazing how our body responds to certain stimulus and how even creates all the response. We don't really think about it, but it's something that it just happened, but you don't think about how it happens, like what makes you turn your hand away from something hot, or what makes you just escape a ball that's coming. It's just everything so quick, and then it's just amazing. And then when I was going through the scholarships, this researchers list, and I read this one and I was like, this is the Upload idea. I have to see this. I don't know what it is, but I want to see it. So yeah, I'm a chemical engineering student at University of Houston. I'm in my senior year. And yeah, I'm just glad to be here and ready to see what is all this about. And I like soccer. And I run sometimes too. So I don't understand American football. If you guys can explain me.
Well, thank you, Anna. So I think you're right. We take for granted many things. And in fact, what you see as behavior is a very little thing compared to what is happening in the brain. Because any millisecond, right, we have choices. We could do many things, and only a few you can see, hear, feel, right? Even as I'm selecting the word I'm speaking, right? I mean, there are so many things I could say, right? But I'm just, I'm selecting those. So what EEG provides is a window to study this brain activity and to learn about the 90% of things that we never see or hear or feel or do, right? So you're completely right. I haven't seen that movie, Upload. I have seen others. But, this concept of brain reading and brain machine interface or brain computer interface is no longer science fiction. And
it is a series, FYI, but I mean, and it is definitely science fiction so far. But yeah, it's good. It's good.
Yeah, and so there's no doubt that all these internal states, movement, emotion, memory, perception, all that, they can be we can access those, right, through neuroimaging, right, and EEG or electroencephalography is just one modality. There's some other ways to read the brain and also to write into the brain, right? That's more complicated, but so welcome to the class. And you can try with soccer. I don't know if the headset will hold, but running probably. I'm also a runner and also a cyclist. I just did 25 miles on my bicycle this morning. It's just good way to wake up my brain. And okay, so Jose.
Yeah, my name is Jose Gonzalez. I'm a PhD student in Pepe's lab. I'm working also in a stroke rehabilitation project at BMI for that purpose. I mainly focus on the electronics and software development. And with respect to my hobbies, I like to play video games and watch movies. But yeah, with respect to the specific purpose of this class, playing video games.
Good. Yeah, we actually, Akshay is going to share a paper that he authored. He was playing Minecraft, by children at the Children's Museum. So that's an example of what things you can do with this headset. Chase, would you like to tell you words about yourself and hobbies and things like that?
Hi everyone, my name is Chase. I'm an undergraduate here at University of Houston. I am studying psychology, and yeah, just super excited to be in this class. Sorry, that's my dog, and somebody is apparently outside. So,
like, what are your hobbies?
Yeah, I think
besides walking the dog, I guess.
Yeah, besides walking the dog, I've recently gotten very into chess, so I've been playing chess pretty seriously.
Oh wow, you have some competition here.
Oh, excellent! I'm always down for a game. I play at least like 3 or 4 hours a day. Play a lot of games actually. If I think about it.
So what's your rating?
Right now,
yeah,
I'm at in rapid, so 10-minute. I'm at 1,000, but that's just because I'm saving my rating. I've been playing Stockfish at around 1,500, and I've been able to tie in most games. So I'm feeling like, yeah, I feel like I think I'm conservatively around the 1,200, 1,300 range. And then my goal is to reach 2,000 by the end of this year, at least in 10-minute. And then I just want to get on the board in 3 minutes. So like 3-minute games, I'm somewhere around like the 1,000 rating. So I'm trying to boost that one up, but it gets difficult. So a lot more opening preparation needs to be done.
Chess is addictive. My youngest kid, he plays a lot online. Andy, what's your score in chess for the quick? Yeah, his score is around 1,600.
That's nice.
And he plays really fast. And I play with him. I cannot do the short. I need time. I do the longer one. No time. And sometimes I'm able to beat him, but these days it's almost impossible. But it's a very interesting game, and it's really addictive. But it's good for development. So that's good. Thank you. So I'm gonna take a screenshot just for the website. So don't move, don't go away, and just to as I said, we have the website, and so this is gonna be one of the first pictures. This is the group.
Can we pause?
Yeah. Okay. So, well, thank you. I mean, those were a very nice introduction. I think we have a lot in common, or something in common, and also some unique aspects. So, so today we're gonna start with the workshop, and I'm gonna handle the Zoom to Akshay. Let me give you access to that. And you should have received my email with more information about what's going to happen this week and in the next 2 weeks. Any questions, let me know. Take notes, whenever you have the headset, feel free to use it. We're gonna meet on Thursday on campus. I will let you know where exactly. I need to check the weather. I need to check several things. But do please, as soon as you can, get tested for COVID. Okay, you can do it. It's for free. You can just sign up online, and you can do it at any of the campuses. Downtown here or whatever. So Akshay.
Yeah. Give me 1 second. You guys can see the screen, right?
Yeah.
Welcome to the today's session. So I'll be the guest lecturer for today. Before I give me 1 second, I'm not able to navigate. Okay. Right. So a few things before we start. These are some of the things that you should expect from this lecture, right? So at the end of the day, if some of this is not clear, please feel free to ask questions. We'll initially cover about what is EEG, the modality that we'll be measuring. So we'll get a sense of what are the origins of the signal. Then we'll get a bit more details about the particular type of headset that I'll be using for the entire duration of the class, get familiarized with it. Now we also will be covering some of the typical artifacts or the noises which will be affected by your brain signals, which will be added. We'll be familiarized with those. And at the last, the session will end with teaching you some of the methods that you can use so that you can clean these artifacts and look at the cleaner part of the brain signal. So this is what we'll be covering in this session. Okay, there is something with the mouse. Okay, right. Yeah, first and foremost, as Pepe said, right, we are going to be measuring a lot of the brain activity in natural environment. So the first question you need to see is what is the best possible image modality that we can do, right? And the reason why we choose EEG, it's because of certain advantages. So the fully research-grade system is going to be like this on the right, what you see here. It has these electrodes with some wires and put on a cap, right? The advantages is that it is non-invasive. So you might be familiar with the system that Elon Musk developed, the Neuralink, which is an invasive technology. You need to break open your skull and put those sensors inside. Now that seems very scary, right? So the advantage of EEG is it's non-invasive. You don't need to do any type of surgery. It's completely portable, so you can carry it wherever you want. That's the biggest advantage of mobile brain imaging systems. It is affordable compared to the other technologies. It has high temporal resolution. By that, what we mean is we can measure changes in the brain in the millisecond time scales. So there are other imaging modalities like MRI or fMRI, things like that, right? So unlike those, we'll be actually measuring the direct electrical activity from the brain. Those technologies measure the blood flow in the brain. So that's some of the key differences and advantages. So this is the type of technology we'll be using. To give a bit more background, as I said, electroencephalography is a way of measuring your electrical activity. Now, I like football, so I'm going to give an analogy of stadium to sort of give how the scaling affects. Now, think of a one-person interview, right? A person is interviewing each individual players, documenting every details of what's happening inside the game. Now, that can be thought of as someone measuring from a single neuron using a system called patch clamps. Now, if I put a patch clamp on a single neuron, I'll be recording very minute details on what's happening in that neuron. Now, we'll be measuring basically the action potential. But then we go to the next stage where it's a busy press room, where they summarize what's happening, the key events that's happening inside the press room. So you won't get the very finer details, but you'll get a very good sense of what's happening there. Now, that can be considered as local field potential. This is still invasive, both these things. You need to put an electrode inside your brain. Now, the local field potential does not measure the activity from a single neuron. It measures from the sum of a set of neurons, but it is still invasive technology. Now, when we talk about electroencephalography, you can think of it like someone is far outside the stadium. So you won't get the entire details, but when something interesting happens, people are going to cheer, people are going to make noises. So you'll get a very good understanding on what's happening inside the brain for interesting events. So we sort of lose some key information, but we do get a good sense of what the technology offers. Now, the reason why I say this is you should be familiar that this is not, it doesn't really tell everything about the brain. There are some limitations. just to have the advantages and limitations described upfront, right? Right. Now,
let me just, Akshay, provide another analogy, one that I tend to use, which is, I think this is good with soccer, but I also like to think in terms of a symphony hall, right? So when you are inside the symphony hall and you focus your attention to the violin, you can really listen for the violin, right? And or for the cello or any other system. You can move your attention to a whole group of say all the strings, right? And then just more local field potential, right? You want to focus your attention on specific, the first violin or the first cello, but the whole group. Or you can listen to the whole orchestra, right? So then you are getting the general information, the sum of the elements, right? And so that will be the EEG component, right? And so that's what another analogy you could use. So basically, there's a trade-off in terms of along different lines, right? One is invasive versus no invasive, right? The other one is time resolutions versus spatial resolution, right? But I want to add that there are signal processing methodologies, some of them adaptive, that allow you to get a little bit more detail that was thought possible nowadays. So, and we will talk, about that some other time. But the technology has improved quite a lot. The signal processing has improved, and so it's amazing the type of information we can get nowadays. So go back to you, Akshay.
Yeah. Okay. Right. So now let's just get into a bit more detail on what's happening, right? Think of an individual neuron and what's happening inside the brain. The communication happens when different neurons talk to each other, and they do it. One means is electrochemical way of conversations, right? So think of a single neuron, and what happens is whenever they talk to adjacent neurons, the fluid out extracellular fluid in around the neurons exchanges ions with the intracellular medium. So there's an exchange of ions between the neurons, through the neurons and outside of the neurons. Now, this exchange of ions is actually the electricity that we talk about, right? It's not really what we talk, the electrons that we move. It's the exchange of ions that is mainly causing the electric field, right? So when the ion sort of accumulates in a particular dendrite, for example, now, is this enough to get a signal outside? Not really, because this is very, very minuscule, right? So the potential created by an individual neurons is very, very small. So if we want to get something to be measured from the scalp, you need many of these neurons to be firing in synchrony, in a parallel arrangement. This is a very idealized situation. Assume that many neurons are aligned in parallel. So this charge would cumulatively build up such that it is strong enough to be picked up from the surface. Now think of the case where and it's not parallel, right? Think of it like in this way. If this is a case, everything sort of cancels each other out, so the potential might not reach the surface. Now, ideally, you want something like this, which is also why they say there is a set of neurons called pyramidal cells, pyramidal neurons. They actually have a parallel arrangements. All the neurons are arranged parallelly. And that is one of the main reasons why people think one of the main contributors for EEG is pyramidal neurons. So when the pyramidal neurons, like thousands of neurons, have sufficiently aligned polarity, it gets picked up by the sensors on the top. Now, this is how it works when the charge gets transmitted through the different layers of the head. These get picked up by the electrodes, which is seen in the figure, right? So the electrodes on the top picks up this voltages. Now, think of the cumulated charge as a single point here, the dipole on the head. Now, when we have a sensor on the top, one of the assumptions we have might be it's just picking up the data from what is right beneath the sensor. Now, that is not really the case. You can think of this dipole or the individual sources as a torchlight, right? It's not like a laser where it's a very streamlined light. It's like a beamed. So whenever there's a source, it spreads to the entire surface through something called volume conduction, right? So this particular source will have contribution in multiple of these sensors. So that's just one thing to keep in mind. So whatever you're measuring will have contribution from different sources, but the major contribution would be which one is the closest to the electrode step. And once these get picked up by these sensors, these are a typical example of an EEG. It's basically these random squiggles, right? Now, it's a bit hard to make sense out of just looking at the data, just from the time series data. One far easier way, one most common thing you can notice is the alpha band activity in the head. Now, alpha band is when the oscillations in the signal happening.
Excuse me, Akshay.
Yes.
Can you explain the source poles again?
Yes. So when we talk about this one, right? When there is sufficient number of pyramidal cells that cumulatively build up a particular charge, which is sufficient enough to reach the top, right? There's a polarity happening, positive and negative polarity happening, right? Across the it's like it's a polarity. It's actually a dipole that is created when you have this. So each individual source, when I call, it's a dipole. So that is what is being projected outside, right? So you'll not have just single source. There'll be multiple number of sources at different areas. So what you're measuring is basically the sum of all these dipolar sources, right? That's what we're getting at the end. So it's not from a single source. It's going to be from multiple locations. That's what we are cumulatively getting at the end. Right. Thanks for the question. Right. So alpha band, what we mean is this is a time series data, right? It contains signal in different frequencies. It's a summation of signals from different frequency components. Now, alpha is a frequency from 8 to 13 hertz range. Now, when I close my eyes in the back of the head, which is the visual cortex, right, which is the occipital lobe. Now, when I close my eyes, there is no visual input. Now, that part, when you do that, the alpha power in that particular region increases, which is seen from this figure. If you see, one on the left is eyes closed, one on the right is eyes open. You can clearly see the difference in this part, right? Like there's an oscillation, clear oscillation here, which is not there in any other part when you open your eyes. Now, this is one of the common ways to see, just from looking at the data, this is one easier way to see a pattern. But many other patterns are a bit harder to see just from the time series. So you need machine learning and other signal processing analysis to do that. Right. And what I have here, I'll try to explain it later too. This is just what is called power spectral density. What this tells is what is the power contained in my signal at each of these individual frequencies. So for example, at 10 hertz, this is the power contained in the 10 hertz power in my signal. But essentially, it's a distribution of that, how much power in each of these frequencies. And the figure on the right, it just shows where the power is highest in the alpha band during the eyes open minus eyes closed condition. It's actually flipped. That's my mistake. Eyes closed minus eyes open condition, right? It's in the back of the head.
Actually, just one, could you go back, just one comment here. If this is power spectral density, the units on the vertical axis would be dB per hertz.
Yes.
Yeah. So there's missing there. And now you are talking about power, then that's correct.
Yeah, it's actually power. Yeah,
power, right? So it's not power spectral, it's power, right?
Yeah.
Now I want to mention that this is a typical example. It's a really a quick test. Close your eyes, you should see alpha go up. Alpha between 8 and 12 hertz, right? Sometimes come to also with beta, which is twice that, right? For example, here you see on this plot, you see two peaks. You see alpha around 10 hertz, and then you see around 21 hertz. You see another peak. That's beta, right? The second peak doesn't always show up, but sometimes it does, right? So when we talk about alpha activity, we're talking about 8 to 12 hertz, but if you are recording from different parts of the brain, say you're recording over the central portion of your brain, which all the sensory motor areas reside, then you also see activity in the 8 to 12 hertz, but we don't call it alpha, we call it mu, right? So we have an alphabet, for this wave, this type of waves, and sometimes the name changes based on the location on the scalp, right? So we will learn more about this, but I want to tell you, right? This is just an example of these things, right? Thank you, Akshay.
All right, so that's the first session. Let's just take a recap, right? So we got I hope you get a basic understanding on what the EEG origins are. Next, we'll try to talk a bit more about the headset that we'll be using throughout this module. So as we saw in the previous figures, we saw a full cap system with a lot of electrodes, right? This is a typical representation of those sensors. Look from the top. When someone looks from the top, this is how it actually looks like. The circles indicate the sensor name and the locations. Now, a typical research study which involving these caps will take at least like 30 minutes to set up, because if you remember, there was a gel, someone putting a gel on the individual electrodes, which is to bring down the impedance. Now, you need to do that for all the 64 channels, and you need to make sure everything is fine, and then connect a lot of other amplifiers, systems, and everything together. So at least it takes like 30 to 45 minutes to get a full system ready. Now that is very difficult, and it's also a bit more harder to move about it. Right now it's easier, but it's a bit more harder, and it's a lot expensive, right? And due to that, there are other alternatives, which are dry systems, where you don't need to put the gel inside. One such example, and it does have a lower coverage. One system is called Muse system, right? This is what it looks like. The advantage of the system, as I said, is it's very fast. It takes like hardly a couple of seconds to put in. The limitation is it doesn't have the full coverage. So if this is the 64 channel, you'll be measuring from these 4 sensors, which I marked in the figure. On the headset, this can be represented by this particular sensor. If you see the silver one here, there are 2 in the front, and the rubber flap, that's for actually flexible sensors. So there are 4 sensors that the Muse can record from. And it also has a accelerometer. So it can measure how the headset or the head is moving using a 3-axis accelerometer. Now this measures the data at 220 Hertz. So every second of the data will have 220 samples of the EEG data.
I have a comment on that. You could go back. So I think it's important to, I know you're gonna say something more about this later, but so for the Muse system, we have 4 sensors, and they are marked here on the figure, right? And on the front, you're recording mainly from prefrontal cortex, right? So this is an area very important for decision making, choice and things like that. But there are also some other type of information that you can get from those sensors. They're very important. That's the executive, right? That's what is telling them what to do. And then on around the ears, on the back, that's a multisensory integration area, right? So pretty much everything we do is takes into account our sensory organs, right? And together with the internal drives and motivation, things like that. But we are really sensing, through our sensory organs and making decisions, right, based on that as well. So with this has said, we can get information about this multisensory integration area, and we can also get from prefrontal cortex, which is pretty decent, right? Also very convenient. It's an inexpensive headset and the other thing is that it has an accelerometer, right? Which basically captures how you move the head. And I'm just going to address that later on, but basically that's important for 2 reasons. One, head movement can come from motor intent. So we intend to move the head in some way, right? To orient our gaze or attention, right? To communicate, yes or not. We use movement not only as a way to, to transport ourselves, but also as a way of communication, right? But also it can be used to study, detect artifacts due to motion itself, right? So that's going to be very important. to combine these. And typically in many experiments, we add to this basic setup also a video camera, because particularly when you are collecting data by yourself outdoors, you want to be able to understand what's the context around you, you know. So when you go back and analyze your brain and movement signals, you understand what was happening at that time. And so we tend to synchronize a portable wearable camera with the brain activity and the motion sensor, so that we have now a context aware mobile brain body imaging system. We call it MOBI, right? So this is the simplest of such systems, and we have studied this system for quite some time. We have published with this system. We understand what are the limitations and what are the advantages of this. And once you start playing with it and getting there, you will understand firsthand, what you can do with it. Thank you, Shay.
Yeah. So yeah, as Pepe said, there's a lot of things you'll know once you have the headset on your hands, right? It's a lot of the learning comes when you actually start doing it on yourself, which is what we'll be doing in the next couple of sessions. I think in Thursday we'll be giving you this headset, and we'll see what are the best practices which we learned also. How do you minimize the artifacts while maximizing the signal? We'll be covering some of them in the next session. Right, going forward. Now we have a good understanding on EEG and also the headset that we're using, but something to keep in mind is when we tell we are going to measure the brain activity, what we are measuring from the sensors is not just your brain activity. Now think of it, right? It's basically a electrode or a metal piece that is put on your scalp. Now it doesn't have any understanding that you're going to be picking up brain signals. Whatever electrical activity it measures, it will pick up. Some of them examples are eye movements. When you move your eyes, that actually also causes, it's like a battery that basically turns off and on. Like when you it's like a discharging when you blink, or if you move the left eyes left and right? So those causes some artifacts. So eye movements cause artifacts. Eye blinks cause artifacts. Then when you clench your teeth, when you are actually scrubbing your cheek, for example, right? Those causes muscular artifacts, which is much higher than your actual brain signal. So the signal gets corrupted by muscular artifacts. You also have cardiac artifacts and many other artifacts. But for the sake of this, we'll be limiting to mainly eye movement artifacts and muscular artifacts to see how they are, right? Now, before we clean them, we should have a sense of how they look like, because we need to understand how they are before we actually can clean them. Now, muscular artifacts typically looks like this, where if you see this is the rest of the signals, but when the muscular artifact happen, it's much stronger and it's actually corrupting most of the channels. This is how a typical muscular artifact looks like. Now, if you remember the figure I showed about the power spectral plot, where the power was changing with frequency, if you have a muscular artifacts at that section, your power spectral density sort of increases, because the muscle activity is mainly focused from 30 hertz and upward. Your brain signal is mainly from 0.1 hertz to like 50 hertz in general. Now there are still higher frequency component, but mostly it is from 0.1 to 50 hertz. However, your muscular signals is from 30 hertz to 500 hertz, for example, right? Just have that in mind. Another artifact is ocular artifact. As I said, the eye movements cause some artifacts. So it typically looks like this. It's actually seen in the other one as well. If you see the blip, it's a clear blip when there is eye blinks, and that's how an ocular means eye-related artifacts. It can also caused by left and right eye movements. It is actually shown by this figure, this pattern here, right? So you should be familiarized with this when looking at the data, so that you know what you're cleaning. Now, I'll at this point, I'll tell you some of the steps that you can do to minimize these artifacts when you're looking at the data. So going forth in your module, you'll be collecting your brain data and trying to analyze it. So you'll need to remove these artifacts and look at maximize your signal in the data. The things that you need to do for this is you need MATLAB 2018. You need EEG Lab Toolbox. It's a particular library which has specialized functions specifically for EEG, right? And I think Pepe sent some attached files. You'll have to download that. And there's a particular script called preprocessing_tutorial.m, which would have all the function that I'm going to be telling here. Now we are going to have a fast-paced section. So if you have any detailed question, please feel free to ask me afterwards. How I plan on doing this is try to give an intuition on why these steps are important, and I'll run by the codes and which are the important sections that you need to focus on. So that's how we'll go the rest of the section. Right. The first step is you need to make sure the EEG lab is added to your MATLAB library. So this, as I said, is a library with a lot of functions related to EEG processing. So once you unzip that particular folder, let's say this is where you unzip the particular library. There are two ways you can load that into the MATLAB environment. One is you can do it to the script, just type add path and particular library. If not, when you open the MATLAB, there is in the home section, there will be something called set path. You can go there and add the particular path in that particular interface. That's one way to make sure the functions are added to your MATLAB path. So when you call a function, MATLAB knows where to look for. That's what we're actually trying to do here. So the first step in analyzing this data is actually loading the data itself. So your particular code will have a section. I would try to label it everything as 1B, 1C to have a tracking with the slides. The first step is loading the data. You load the data from the Muse. So here again, we assume that we have the data, everything saved. On Thursday, I'll show you how the data can be saved. That section will be done on Thursday. I don't want to give too much details right now. So here we assume that we have the data and we are just showing how do you take that into the MATLAB environment and remove the artifacts. The first step is loading the data. You just need to rename this particular file with the actual file name and then run this section. So that's going to load the MATLAB variable into the MATLAB environment. Now, MATLAB also needs to understand what are the different type of channel. The EEG lab function needs to understand what are the different channels. By this, what I mean is, if you remember the previous slide where there was a circle and we marked the TP9, AFC, certain circles, right? So it's just indicating which are the channels that each of the columns represents. It's just for the EEG lab to understand this. So the first part is loading the data and then just loading the channels. You just run this lines of code. Only thing you need to change is this particular section here, the file name. Right. Typically, what we do is we downsample. Now, this is mainly due to computation requirements. For example, as I said, EEG is mainly from 0.1 to 50 hertz. Your Muse headset samples at 220 hertz. Now, if you don't really need like that much 220 hertz, okay, let me just back off here a second. If you're not familiar with the Nyquist criterion, I'll just try to explain that first, right? What Nyquist criterion says is, if you want to study a particular frequency of interest, you should be sampling it at least twice that frequency that you're looking for. For example, if I want to study 30 hertz, your sampling rate should be at least greater than 60 hertz, right? So that's what Nyquist criterion shows. Again, to intuitively explain this is an example simulation I gave. It's a 10 hertz sine wave simulated at 1024 hertz. Now, if I do downsample it to 120 hertz sampling rate, it still captures pretty much the entire signal. If you do 32 hertz, you still get the frequency component preserved. But if I do 16 hertz, now half of that is 8 hertz, which is lower than your actual signal content. Now the signal is completely distorted, right? So it's always good to have, always make sure your sampling rate is twice the frequency you are trying to capture. Now in here, you downsample it to say 110 hertz just for computation purposes. This is not required, but just as typically we try to do that for computation purpose. Now, for example, if I'm taking like 1 hour of data, right, that's going to be a long recording, and the processing time the computation might be very extensive. So it's always good to downsample it to say 110 hertz or something. This is a code. It's very simple. You just need to mention what is the sampling rate that you need to resample it to. Just put it as 128 to 110 here. The next step is high pass filter. So before we do this, let me try to explain why this is important. When we wear a headset, especially think of we going outside in the open settings, right? You tend to sweat, or there are some changes in the environment, right? That can cause differences in the impedance in your sensors. Now that causes a slow change or a drift in your EEG data. It's an exaggerated version here, but it's actually a real data. So you can see it basically amplitude basically drops over a period of time very slowly, right? So we want to remove that particular noise. So we do high pass filter. What high pass filter does is, as the name suggests, it passes everything higher than that particular cutoff frequency. High pass, it removes everything below that cutoff frequency. So if I say I'm making a high pass filter at 0.1 hertz, I am preserving every signal above 0.1 hertz. I'm removing everything below 0.1 hertz. So at the end, you just basically remove this drift, you get something like this. So that's what typically the first steps I try to do to remove this drift artifacts.
I want to add that there have been studies about what's the best high pass frequency cutoff, right? Now, Akshay, could you go back to your 1/f curve that you showed before? Yeah, so that curve, power versus frequency, right? So you see the power goes down with frequency. In fact, it's a power law, right? And that's typical of EEG, right? Now, if there are artifacts, say, assume that there is, we have muscles here on the head, right? When we do faces, or when we move or blink, there are some muscles here that contaminate the brain recording, and that means that this higher frequency noise that corrupting your brain signal that has 1/f. So the 1/f is going to go away, right? Because you're going to see an increase. So it's going to go down, and you're going to see an increase due to the myoelectric activity from muscles, for example. Or you have motion, right? That's going to produce some low frequency changes in the 1/f. So these artifacts will change the 1/f. Now, something that we know is that below, so there are, as Akshay mentioned, different frequency bands, right? We talk about alpha, 8 to 12 hertz. In fact, there are, delta, which is, between 0.1 and 4 hertz. And some people actually consider delta to be from 0 to 4 hertz, although I think that's incorrect. And there is some debate about whether it starts at 0.1 or 0.3 or something like that, right? Some people, take it from 1 to 4 hertz, for example. So there's a little bit of debate on that. But delta is a low, it's low cortical potential, right? And then you have theta, which is 4 to 8 hertz. Then you have alpha, 8 to 12. Then you have beta, 12 to maybe 24. And then low gamma from 24 to 32, 40 hertz. And you have high gamma, that goes to maybe 100 hertz. I think the highest frequency you can record with EEG is around 100, 150 hertz with really careful recordings in a specific situation, right? So the bandwidth is not that great, right? But there's a lot of information in what we are recording. Now, below 0.1 hertz are the ultra slow cortical potentials, right? These are things that you see during sleep. These are things that are seasonal, that are changing based on the context. And that you can see from this plot, the things that are close to 0 frequency, 0 hertz, has the highest power, right? And these are the most, the frequencies that are more noisy, right? So you don't want to compare something that happens, say, at 30 hertz, with something that's happening at 0.1 hertz, because these are these are big difference, right? And so there are studies that suggest that you want to detect, for example, movement intent from EEG. We know, we actually discovered that movement intent is represented in the fluctuations in the amplitude of slow cortical potentials in the delta band, right? So I said a lot of things here. So let me just go back and explain. So slow cortical potential could happen between 0.1, 0.3 hertz to about 4 hertz, right? These are slow rhythm. They are not ultra slow, but they're slow, right? They tend to happen over, cortical area related to movement and sensory information, central areas of the scalp. Although I had to say that behavior with EEG is best detected over the whole scalp, right? There's a neural network, right, that different parts of the brain are working together, because any task requires a lot of information. You can consider, for example, pointing to a target, right? You need to know where is the target. Normally, visually, you need to know where is the current position of the arm, the hand. Normally, that's proprioception. Or you can look at your arm too. Or maybe the target is auditory. Maybe there's a sound that you want to aim to. And so there's a sensory integration with movement, right? And so the information about movement intent, is represented in the change in the amplitude of slow cortical potential in the delta band, right? You can think about a radio in your car that has AM, amplitude modulation, and you have FM, frequency modulation, right? And when you are recording inside the brain closer to the neuron, local field potentials, right, or maybe single unit activity, normally you get this information in the frequency domain, right? Frequency modulation. You see changes in the frequency or the amplitude of particular power. But you're using EEG, your best bet is to use actually AM, amplitude modulation, right? Which means, you need to know which station to tune to, using your AM, right? We know that is delta. So our local station for movement intent is in the delta, is delta station, right? But also there's beta, and there are some others that also convey some different type of information. So it's complicated. I'm simplifying here a lot, but again, we want to remove the very slow cortical potential, which are less than 0.1 hertz, right? Unless you're interested in those, right? And some people look into those rhythms because they want to, they're making investigation about, for example, consciousness, right? Or changes in internal state over a long period of time, or maybe looking at plasticity of the brain during sleep, things like that. So there's information there, but it's very hard to get. You need long recordings, to record, to get a frequency, say a 0.1 hertz, right? You need at least 10 seconds of data, at least, right? You normally need more than that, and then you go lower than that, you need minutes, and so you can see it gets complicated. Anyway, all of these we will have an opportunity to test ourselves, because you can look at all these frequency bands, right? Going back to you, Akshay.
Yeah, thanks, Pepe. Yeah, just to add that point, right? Pepe said about different bands, right? So if you want to compute the power in any of this, the sort of suggestion that we say is try to get at least 3 oscillation of interest. So if you're looking for 4 hertz, your window size should be able to capture at least 3 times of that. So it should have 3 times 4 hertz oscillation. Like just to keep that in mind. Right then. Right then, so right now we remove the slow drift. The other is low pass filter. Now I talked a bit about the muscular artifacts, right? So it's, as I said, it is from 30 hertz and above. So it's mainly from 50 hertz to 500 hertz, and we are sampling at 120, I think 220 hertz. So you actually have signals from 50 to 128. Because it's a dry system, and because we are walking a lot, you can really expect a lot of artifacts, muscular artifacts to be present in your data. So one way to remove that is you low pass filter I'd say 30 hertz or 49 hertz, right? So you remove large part of the muscular artifacts. Considering this to be walking, there's a lot of walking involved. I would say maybe 30 might not be bad. Just look at the data. That's the best thing. Always look at the data, see whether it's too much corrupted. If you feel like you can appropriately do. Again, to explain, just the opposite of high pass, you're actually keeping everything below low pass. It passes everything lower than the cutoff frequency. As an example, this is simulated data with 2 hertz, 10 hertz, and 80 hertz. And if I do a low pass filter at 30 hertz, everything 2 hertz and 10 hertz is less than that, so it's preserved, but 80 hertz is higher than that, so it's basically removed from the data. That's essentially what a low pass filter does. Similarly, if you low pass filter at say 30 hertz, you're going to remove majority of the muscular artifacts, but it's going to retain the key EEG informations present. Again, the same step. The only thing you need to check for is the cutoff frequency here. That's the only thing you might need to change depending on the data. This is very important. Whenever you do anything in MATLAB, always do sanity checks. This is very, very critical because sometimes your columns might be flipped, right? So it might give you the wrong answer. So it's always a best practice to before and after plot the data and see what's happening. This is the same graph we said. The function will have a code for doing this too. So what you need to check for is, say we have a cutoff like high pass filter cutoff at this line, right? So if this was working, if you see the row original data, there is power before that particular cutoff in the original data. But in the high pass filter, if you see, it actually went out to like minus 60 dB. So it's cleaning everything less than that cutoff, but it's preserving everything else. Now check the high pass filter compared to the low pass filter. So this is the cutoff for the low pass filter. So it's going to remove everything after that. So only in the low pass filter, it is going down, but in the high pass filter, it's still up, right? So these are just a few ways to double check that what you're doing is correct. So always run this whenever you run this particular analysis. There's a code in the script that can do this for you. Next step I try to do is removing bad segments. So because these are, we have a lot of movement, some of them would be very big artifacts. So it's always good to remove them. If you see like very big artifacts compared to the data, you can just remove them. How you can do it is, after doing that low pass filter, write this function called EEG-lab redraw. What it does is, it is going to update this GUI. There is a GUI that actually pops up with the latest value of the low pass filter data. And then what you do is go to tools, click inspect or reject data by eye, right? Click this section, and this pop-up window will gonna show up. And what you can do is, if you look at the data, if there are instances with a lot of artifacts, you can just select and drag, and the blue section like this will come up. You can select multiple locations, and once you selected this, you can just click reject, and that section is going to be rejected from your data. That's one way you can remove the bad segments from future analysis, right? That's one way to do it. Then there might be instances where some of the channels, when you fit this headset, when you're walking, there's some chance that some of the channels might be not be making good contact with your scalp, right? So here, this is an example on how to identify when you plot the data. So the 46 channel looks very different from the rest of the signals, right? That's one way to see that it's pretty bad. So what you need to do is make sure you remove that signal before doing the future analysis. This particular line will help you do that. Instead of 1, you just put 46, right? And just run this line. Right now, I put it under a if false condition because typically your data will be cleaner, so you don't want to simply run it. So if you really have a bad channel, just uncomment this and run this particular line, right? So that removes the bad channel.
One question, Akshay.
Yeah.
The number of channels depends on the type of headset.
Yes.
Will this function recognize what kind of data set you have?
Yeah. Yes. So that's why we are loading the channel location file. There's an eeggs.cd file there. So you just need to load it. So the EEG lab structure will know how many channels it has. All the information will be there. So make sure you to load that information there.
Yeah, because right? For the Muse would be 4 channels.
Yeah, it's going to be.
I don't know if you plot also the acceleration as a channel or not.
Not in here, no.
Okay.
Right. So so let's just take a stop here and just try to understand what we did, right? Uh, Again, the purpose of this is we are going to be collecting your own data. So there's going to be artifacts in your data. We're trying to minimize those things. There are a few things we already removed. We removed the slow frequency drift from the data. We have a function to do that. We did low pass filter to minimize the effects of muscular artifacts. We do low pass filter at 30 hertz or 49 hertz. And if there are segments of data which is corrupted, you figure out a way to remove that. And if some channels are bad, you basically remove that as well. So these are steps we right now did. Now, there are going to be eye blinks every now and then in your data. You're going to be blinking very often. And there might be some blips in your data, which also needs to be removed. Manually removing all of them will lose a lot of data. So instead, what we can do is something called artifact substrate reconstruction. I think we are a bit running behind, so I'm not going to give full details on what it does. It actually is, think of it of a way of removing any sudden burst in the data. Just assume that this is a sudden burst, which you don't really want in a data. ASR basically does that. And it is based on an approach called principal component analysis. And if you're not familiar with this, just to give an intuition, right, what PCA does. If I ask you to explain a teapot, for example, right, I can take multiple images from different angles, and none of these images might not be explaining the teapot individually well, but there's a particular angle at which if I take a photo, it explains my teapot really well, right? So PCA is in some sense trying to find which direction to look at the data, but which covers the maximal information, right? So the particular approach is based on PCA. The only thing you need to take care of is to remove this sudden burst in the data. And this is a line in the code that you need to, this is the only thing that you need to worry about. And the only parameter for the sake of this class you need to worry is this one, burst criterion 100. This is very high. You need to keep it at like start from 10, go down 5 or 15, 20, try different variations. And I'll show you why this is very important, right? This is one example data I collected from the Muse. If you see the spikes, these are artifacts that you don't want from the data. This is eyes closed condition. This is eyes open condition, right? That's indicated by the signals. Now, if you see here, this is a clear artifacts. As I said, the muscular artifacts are from very high frequencies, right? It's extending to 40, 30 and everything. Now, if I click a proper threshold, right now it's at 10. You see the cleaner part where there is eye closed, there's no artifacts there. The entire signal is preserved completely, right? If you see here, there's no reconstruction happening. The frequency distribution is also the same. But and even when there is artifacts, see, these things are reconstructed. It's removing those sections. However, if I do a bad threshold, if I do a very extensive threshold of 3 standard deviation, now you can see even the cleaner part is reconstructed. The data is basically killed in most parts. Even when there is no artifacts, it's basically removing that section. So this is very important that you find the appropriate value. For example, if I do 50, it's not really cleaning my muscular artifacts here, right? So the only thing you need to check is make sure you try it at different values, different standard deviations, and see which standard deviation works best for your data. So it's never a single answer. You just need to try a few things, right? Yeah, that's another step. These are some of the steps that you need to do. That as Pepe mentioned, it also has accelerometer. So you can look at the accelerometer data and see instances where they are bad and remove that segment too. That's another way we will do it. One other key element you need to take away from here is there is no single way of cleaning EEG data. That's a reality. The different study, different data set will have different type of data. For example, Pepe said about the frequency that you're interested in really drives the pre-processing. There is something called Wershaft potentials. So it is actually a preparatory signal when you're actually planning on moving your hands, for example, and things like that. There is a slow moving cortical potential, which basically goes down, right? That is in the range of 0.1 hertz, etc. Now, if I do a high pass filter at 1 hertz, you're actually killing the key information, right? So depending on your study, your parameters will change, etc. So these are some of the steps that you need to worry about. Now, as a recap, I want you guys to sort of answer these questions right now. If you have understanding on what EEG is, the signal, what are we measuring, right? We are measuring EEG. If you have a good understanding on that, that's good. And the other question is, how do you measure it? We are going to be using the Muse headset. We basically introduced it. We know what it can measure, what are the signals that it can do. Now, the other thing is we talked about certain artifacts. You should know what are the expected artifacts you can, or the noises that you can expect from your data that you collect. Then we also discuss some of the approaches. So ask yourself if you understand the general approach. Now, we did cover a lot of things here. Some of them might be over your head right now, but that's fine. Check the video again. You have the code. Use this as a reference to see what are the things that you need to modify and change when you're running your own data, right? I would say if you're stuck at this, watch the video, check. I've commented the code as much as possible. This is to help you remove some of the artifacts and help you with the data processing, right? So just revisit this video and check the code and try it yourself. Now, if you say yes to all of these things, I think the purpose of this lecture is good, and we should be good for the next session, right? And right now we ran through a lot of the things, but if you want to go into details, these are some very excellent resources that you can go through on EEG and things you can go over, right? Right. So yeah, if you have any specific questions or anything in general, just let me know. That should be all from me for now.
Yeah. So thank you, Akshay. So yeah, so these are good references in case you're interested in going a little bit deeper on what it means, right, to record EEG, and you have to read it for tomorrow. I mean, these are some references that you might be interested, reading critically, in the next weeks, the best way to learn EEG is by doing it, by experiencing the different type of artifacts, and given that you're gonna be collecting data from yourself, I think that's gonna be even more interesting, right? Because you can collect under different conditions. I mentioned, walking, sitting, reading, playing chess, running, maybe, any, or you can just put, the headset and go about doing your day for the whole day. I don't know. Well, what is the battery time for this?
They say 3 hours.
3 hours. So
Yeah.
you need to recharge it, right? But 3 hours is a lot of time, right? So once you collect that data, then the first thing I would recommend, is you can compute the power spectral density, over frequency to understand this 1/f power law, right? And see whether that is followed for the different type of conditions that you record your own activity. That's just an example, general example. In any case, we will handle these Muse headsets to you. I mean, Chris already has one, but we will handle to whoever doesn't have one. For that, we will need you to complete a form. I don't know, Akshay, you have the form. You can send them so that they can get them filled out. And because we need to have we need to account for each headset, right? That's a regulation. And then we're gonna meet. I suggest to meet next Thursday, on Thursday at 10, between the building 1 and 2, right? So you remember there's a birthday, there's a culture, right? As you walk from one building to the next. So we can meet in that area, and then we can distribute the headsets. We can show you how to set them up yourself, and then we can go around and do different type of recordings, so that you have some initial database, and then we can use that to use the scripts, MATLAB scripts that Shay has provided, to start looking at the signals. And then you can take it home and then get more data, right? The idea is that, by next week, do we have a better understanding of the headset? What's the best way to position the headset on your head? Because you need to adjust it. And and have an understanding, how to turn it on, how to stop the recording, so on and so forth, right? And then we're gonna have some session where we're gonna process the data based on this script, right? And what you can do with the data is really open, right? I'm gonna send you a paper that we published at Chase, the first author, that used the headset to study children that were playing Minecraft, right? Then, as another example, we just published a paper in Frontiers in Human Neuroscience. It was an experiment that we conducted with a student from the creative writing program at UH. So the students use the headsets, during class, and also when they were, walking in different parts of Houston, and then writing about their experience, right? So we published it's a short paper, and so you can have an idea about, the methodology, type of question we were asking, and what we did with the signals, and how they look like, and things like that. So we're gonna send those two papers, and you should focus only on the background. What was the experiment about, and then the method, how we run the experiment. And then later on, we will look at the analytics, but let's take one step at a time. So any questions so far? Now you can remove the slides, Shay, and let's go back to, yeah. So yeah, Chris.
Does the Muse device have a way to save the data?
Yes, so we'll be doing it on Thursday, the actual saving. And so here, this lecture assumes that we have the data, but on Thursday, I don't want to bog. I mean, already this is too dense of an information for someone who is new to it, so I don't want to give too much information right now. So we'll be doing it on Thursday, how to save the data. Again, I want to say something. A lot of the information that we talked about might be a bit hard for if you're hearing for the first time. A lot of the terminologies, jargons might be different. So don't worry. The only thing is, we just wanted you to be familiar with the script that we provided, right? There's a script. We want you to know what are the things you need to modify to work with the data. I mean, these are things you learn on the fly. We learn as we go, but this is just so that you have, what are the tools you have, what to look for, where to look for, just to get familiarized, right? But yes, we'll be detailing how to save it on Thursday. And Chris, I think you should get the older headset too. I think that might be better to have it systematic, maybe.
Right. So we will distribute not only the headset, but also a tablet, a small tablet, so you can see the recordings in real time as you go around. I don't know if we have cameras. It's certainly necessary, because we will mark what we are doing on that. But
don't we have cameras?
Yeah, maybe we can do it for some of the other sessions, because I don't want to put everything together from day one, otherwise it's going to be too much. So we can add that later on, get more sophisticated, later on. So yeah, so we're going to meet between, building 1 and 2 next Thursday at 10. We're going to bring the headsets. Don't forget your mask, right? Try to get COVID tested, right? It's free, and it's no pain. It's really fast. Then you're welcome to read one of the suggested readings. Again, this is not for next class. It's general information that is going to be helpful to you at some point, right? And do not hesitate to ask questions. So, Alex, could you tell us a little bit about the website in the last minute? How do you want to run that? In terms of, I have a picture of the group, so that would be the first one, right? And I can write something to start, and then as you start playing and reading, or having ideas, you're welcome to write some succinct text and maybe a picture, a video clip, and just upload it, right? So, Alex.
Yeah. I mean, you send me anything you have, send me pictures you have. I have the framework in place on GitHub pages.
Right. Do you have the link for the website that you can share with everyone?
Well, yeah, I mean,
or send it to me. I want to make, I'm going to review it.
Yeah.
And then I can share it, and I think it would be nice if everybody could just go there and make, provide content, right? And going through you, because I don't want you to be overwhelmed by, a lot of information. So if we can set it up in a way that we can share the password or create different passwords, I don't know if we can do that, but we can share a password within the group. Because, it's going to be like, you're going to have a notebook, right? Or, you're going to have your note, but this would be, a filter note where any thoughts, comments, experience, challenges, questions you can put there, right? And to document that, okay? So we will talk about that, Alex, and yeah. Any other question?
Yeah, I have a question.
Yeah.
Where are we gonna meet on Thursday?
Well, yeah, you remember on the ground floor between building 1 and 2, there's this sculpture, right, of a bird, or I don't know what's that, or the between building 1 and 2.
Between the two buildings, yeah, I know.
Between the two buildings, we're gonna meet there, right?
Okay.
We're gonna be outdoors wearing masks. We're gonna have the headsets. We're gonna exchange that for a form, okay? And then we're gonna go and do some recordings and answer, questions, and get ready for the following week. We're gonna do this test. Any other question?
Yeah, Professor. I won't be able to join this Thursday in person.
I know that you're gonna be away this. So what you can do is before either if you can meet with Akshay on Monday that's February what 2nd. February 2nd. I think we have. Let me just check the calendar so I don't give you wrong information. Well, yeah, February 2nd we have a Zoom guest lecture, right, by the curator. So we can arrange a meeting with you to give you that headset that day or that week. But you really need to get it that week because you need to be knowledgeable. You need to be able to use it on the 9th, right? So you can read papers. And then ask questions just to catch up, right? But so let Akshay know when what would be a good day and time to give you that, okay?
Okay.
Otherwise, will be this Thursday at 10 a.m. Any other question? No? Okay. So, well, thank you. So, it's 11:33. You have any follow-up questions? You are welcome to email me or Akshay. Akshay is the TA for these workshops. Okay. Again, right now we have this color field. They're gonna be more. So you're interested in participating in the other ones, let us know. Certainly, next week. Maybe you try the first one and then make a decision, or that you want to do it. Just let us know so that we can reserve your place. Okay. All right. So thank you. Have a nice day.
Thank you, Professor.
Bye.
Yeah.